Is it visible?
Yes, sir.
Okay. So biopotential, today's lecture based on biopotential and bioelectrodes. So actually, human cells, these are too small in size. Okay, they can be seen only through a microscope. So each cell, it has generally one nucleus and an outer plasma membrane. All of you know that. So various signals are actually generated by human body in the process of carrying out various functions. Now these generated signals are basically bioelectric potentials, which relates with different nerve, muscular activity, heartbeat, etc. So basically, bioelectric potentials are consequence of some chemical changes in the associated cells. Okay, and the muscle cells that can be excited chemically and mechanically to produce an action potential that is transmitted along their cell membrane. Okay, so what is bioelectric potential? So actually, the ionic voltages, these are produced as a result of the electro chemical activity of certain special type of cells. uh, these are known as bioelectric potentials. Okay, so an electric potential that is measured between points in living cells, tissues and organs, and which accompanies all biochemical process. So it basically describes the transfer of information between and within cells. So in with this context, transducers are used to convert this ionic potential into electric voltages. Now these signals that can be measured and interpreted usefully in a meaningful way to aid the physician in diagnosis and treatment of various diseases. Okay, so certain systems of our body that generate their own monitoring signals while carrying out their various functions. Now these signals convey useful information about the functions they represent. Now these signals are uh, called actually ionic voltages. Okay, so and basically these are bioelectric potentials. So bioelectric potentials, these are generated due to nerve conduction. These are generated due to some brain activity, heartbeat, muscle activity, etc. Okay, so special type of cells such as nerve and muscle cells of human body, these are encased in a semi-permeable membrane. You know what is semi-permeable membrane? A permeable membrane, actually it permits some substance to pass through while others are not permitted. Okay, and this concept has been established through experiments. So basically the ions inside the membrane, these are called internal cell fluid, and the ions outside the membrane, these are called actually external cell fluid. So when uh, this semi-permeable membrane is in normal condition, the sodium ions remains outside the membrane. Okay, and in the normal condition, what I mean, the sodium ions cannot pass through the membrane. So what happened, the potassium ions that can pass through the membrane, as permeability of potassium ions are too high as compared with the sodium ions. So if the membrane is stimulated or excited, stimulated if the so if these membranes are stimulated or excited, what I mean, the characteristics of this membrane also changes, and therefore the sodium ions can enter into the uh, cell. Okay, and once the membrane is stimulated, all the sodium ions can enter the membrane. At the same time, the potassium ions try to leave the cell. Okay, and it creates a balanced condition. So the bioelectric signals produced in our body that are due to basically coordinated activity of this large group of excitation cells. Okay, and different this type of, so this bioelectric signals has different amplitude and frequency range. Okay, so don't need to consider that. So you just remember that it has some a range, because some bioelectric potential, like it is, if it is related to heart activity, it is related to brain activity, muscle activity, and for this type of individual activity there is some amplitude and frequency range. Okay, and this range can be recorded by different machines, different procedures are available, like ECG, EEG, EMG, etc. So what is, move on to the next topic, what is resting potential, resting membrane potential, or resting potential. So we know that some of the type of cells of the body, these are uh, encased in a semi-permeable membrane, which allows some substances to pass through the membrane, whereas others are not allowed to pass through. And we also know that these cells of the body are surrounded by body fluids, which are conductive solutions containing different charge atoms, that is ions. Okay, now the prominent ions are uh, sodium, potassium, and chloride. So the membrane of the cell allow entry of this potassium and chloride ions, whereas blocks the entry of the sodium ions, like in this figure here. So various ions seek a balance between inside of the cell and the outside of the cell. Okay, the sodium, actually sodium is unable to penetrate the semi-permeable membrane. So this results in unbalance of ions concentration and electric charge. Now, this concentration of sodium ions inside the cell becomes much lower than in the intercellular fluid outside, and the sodium ions are basically positive. So therefore this makes the outside of the cell more positive than the inside. Okay, in an attempt to balance the condition, the electric charge, the additional uh, potassium ions, which are positive, it enters the cell, causing a higher concentration on the inside than the outside. Okay, and in this way the charge balance, uh, somehow it is balanced. So equilibrium is achieved with a potential difference across the membrane. So the inside of the cell is negative and the outside is more positive. So this membrane potential called the resting potential, and this potential is maintained until some kind of disturbance is caused to upset the equilibrium. Okay, and the resting potential, basically it ranges from minus 60 millivolt to 100 millivolt, minus 100 millivolt. And remember, a cell that is in the resting state is said to be polarized. Okay, this is the polarized condition. Okay, so that is the resting potential. Now another potential has there, that is called the action potential. Okay, look at this figure, action potential. Now in so basically the excitation of a section of cell that can be by using the flow of ionic current or any externally applied energy. Now this excitation of a section of this cell that causes change in the characteristics of the membrane, which allows some of the sodium ions to enter. Now this movement of sodium ions into the cell further accelerates the entry of the sodium ions. Now this leads to rush of sodium ions inside the cell, whereas potassium ions try to go outside but are unable to out that fast. The net result is slightly higher potential inside the cell. Okay, and this potential is basically known as action potential, which can be about mm, plus 20 millivolt approx. Okay, and we can also say that cell which is excited gets depolarized and leads to action potential. So when it is in resting potential mode, it was polarized. Okay, but when it goes in action potential, so it will be depolarized. So the process of changing from resting state to action potential is called depolarization. Is it clear? Is there any confusion, any doubt?
No, sir.
Okay, thank you. So move on to the next slide. Propagation of action potential. So first understand this graph. Okay, so when the rush of sodium ions through the cell membrane stops, a new state of equilibrium is reached, and the ionic currents that lowered the concentration of sodium ions, it was not present there to balance condition, to create the balance condition. So the membrane behaves in its normal condition, that is sodium ions are not allowed to enter inside from outside. Now at this stage what happened, sodium ions are quickly transported from inside of cell to outside of cell, and this active process is known as sodium pump basically. Okay, so once all sodium ions are pumped outside the cell reaches its resting potential mode, and the process of uh, change from action potential to again resting potential is known as repolarization. Okay, so this position is called repolarization. So very a little it is known as about the reason of sodium pump, but it can be to be balancing effect after ionic currents are removed. So it is so you can see it in this figure. The time scale depends on the type of cell producing the potential. So nerve and muscle cells may have 1 millisecond duration, whereas heart muscle duration may be high, uh, more than this, approx 300 millisecond. Okay, so here you need to remember that thing, that after repolarization completion, resting potential is named after potentials. It is called as after potentials, which reaches resting potential slowly. After this, it goes into resting potential slowly. Okay. So when so once a cell is excited and generated action potential, ionic currents begin to flow. Now this process excites neighboring cells or adjacent areas of the same cells. Okay, and All of you know that a nerve cell with a long fiber may have action potential over a small segment of the fiber, but it is propagated in both directions from the origin point of excitation. So the rate at which an action potential moves down a fiber or is propagated range cell to one cell to another cell is called the propagation rate. So in nerve cells, the propagation rate that is ranges from 20 to 100 meters per second. And the propagation rate of heart muscles cell is slower compared to this. That is 0.2 to 0.4 meters per second. Okay. So when this action potential propagates, ionic currents, so what a bit in this process, ionic currents flow if a cell is excited and it generates an action potential. But this process excites the neighboring cells, as I have told you earlier. In nerve cells, only a small portion of the fiber undergoes a transition. It may be noted that the membrane lying ahead of this active region that is polarized, and this active region is depolarized, whereas the membrane behind the active region is repolarized membrane. So the charge distribution has closed this currents, these path currents, and the flow of these currents depolarizes the membrane in the region ahead of the active region so that it becomes activated. At the same current, at the same time, the same current pattern flowing behind this active region is unable to re-excite the membrane in reporting state, whereas the process is actually self-exciting state. And now the active state of the membrane is remaining only for short duration of time, and after depolarization the membrane uh, repolarizes completely, and this is the way the action potential propagates along the length of the fiber. Okay. So synaptic transmission, is it visible, this slide? Synaptic transmission.
Yes, sir.
So it is actually it is the process by which signaling molecules, these are basically these signaling, what is signaling molecules? These are actually neurotransmitters. So these signaling molecules are released by a neuron, the synaptic neuron basically, and bind to and active the receptors of another neuron, which are called the post synaptic neuron. Now neurotransmission, that is very essential for the process of communication between two neurons. Okay, and these are reliance on certain factors like the availability of the neurotransmitter, so that the release of the neurotransmitter by exocytosis method can be there, and the binding of the postsynaptic receptor by the neurotransmitter, as well as the functional response of the postsynaptic cell, and the subsequent removal of the deactivation of the neurotransmitter. So these are the different factors on which this synaptic transmission depends. So when these action potentials, which I have told you earlier, when this action potential reaches the end of the action and the action terminals, it creates the membrane sacs called vesicles to move towards the membrane of the action terminal. Now the membrane of the vesicle basically fuses with the membrane of the action terminal, enabling this vesicle to release some of its contents into the synaptic space. So when a nerve impulse it reaches the synapse at the end of a neuron, it cannot pass directly to the next one. Instead, it triggers the neuron to release a chemical neurotransmitter. And this neurotransmitter actually drifts across the gap between two neurons. Okay, and there is there are certain ways there are mm, by which this neurotransmitter is deactivated. One is the reabsorption of the neurotransmitter into the neuron through channels in the membrane. Another is the destruction of the neurotransmitter into a substance which has no effect on the receptor channel with special chemicals called enzymes. Another way is the diffusion process by which the neurotransmitter becoming detached from the receptor and drifting out of the synaptic cleft. Okay, so this is basically synaptic transmission. Coming to the bioelectrodes, so bioelectrodes are this function. So bioelectrodes function as an interface between biological structure and the electronic system. Okay, interface between the body and electronic measuring device that is very much required to measure and record these biopotentials. Now, this interface function is achieved through electrodes. In order to measure signal current as to flow through the circuit, therefore electrodes must have capability of conducting a current across the interface between the electronic measuring circuit and the body. Okay, and these electrodes basically uh, carries out a transducing function where current flows in the body by ions along with the current flow in the electrode by electrons. So in the bioelectric potential which are generated in our body, these are ionic potentials produced by ionic current flow. Now measurement of these ionic potentials actually it requires conversion into the electronic potential. Now, according to the development of modern noise-free, different and stable measuring devices, those are available. Now these devices are used to convert ionic potentials into electronic potentials, and this device actually calls electrodes. So the design of electrodes, these are dependent and understanding bioelectric potential in general and measurement of different like pH, for pH measurement, for oxygen pressure measurement, for carbon dioxide pressure measurement of blood in different ways. So this uh, everyone mute yourself. So the measurements of this ionic potential and the measurement of electric currents in body need some interface between the body and the electronic measuring instrument. Now this function is achieved through the biopotential electrodes or bioelectrodes. And this potential measurement is done through the current flow in the measuring circuit for some time over which the, the measurement is done. Basically, in ideal condition, what happened, this current should be very small, and electrodes should have the capability of conducting the current across the interface between the body and the electronic measuring circuit. Okay. And actually this electrode basically carries out a transducing function, since current is carried out in our body through ions, so it is carried in the electrodes and its lead. Okay, it has some leads, so lead were by electrons. And every electrode, it has a transducer to change an ionic current into an electric current. So that is the basic idea about the bioelectrode. So okay, so the electrode and electrolyte interface, that will be discussed next day. So if you have any confusion till now, that's all for today. If you have any confusion, you can ask me now. Do you have any confusion?
No, sir.
Okay, so